Hello, my name is Misael Prieto, and my journal club presentation will be about human gut bacteria and their association to neuropsychiatric disorders. Thank you. Bluff. Bottom line up front. So we know that the gut microbiome plays a role in neuropsychiatric disorders. This is because other research has found that bacteria in our gut can sometimes create neurologically active compounds that send signals to our brain and tell us what to do or affect our emotions. But this study specifically investigates the effects that Escherichia coli and Lactobacillus mucosae have on neuropsychiatric disorders in mice. So E. coli was found to exacerbate neuropsychiatric symptoms. This is because it can cause gut dysbiosis, or it can cause us to have a negative shift in our um, gut microbiome. L. mucosae was found to sometimes mitigate these symptoms. In the last slide, I mentioned that the gut and the brain can communicate, but there is a research term for this, and that's called the HPA axis or the gut-brain axis. This is communication between the microbiota in our gastrointestinal tract and the CNS or our central nervous system. This happens when microbiota in our GI tract create neurologically active compounds such as epinephrine or other hormones that can regulate or affect our mood. I also previously mentioned gut dysbiosis, which is an imbalance of the microbial community in our GI tract. Now, this is associated with various neuropsychiatric disorders because when we have gut dysbiosis, they're more likely to send neurologically active compounds that create negative emotions in our brain. Now, why do we need more research that involves neuropsychiatric symptoms and gut dysbiosis? Well, after the COVID-19 epidemic, there's been a prevalence of neuropsychiatric disorders across the world. This research shows a potential for a microbiome-based intervention based on probiotics and dietary management for improving mental health. A lot of current neuropsychiatric disorder medications are invasive and harm our microbiome, so creating one that could potentially help it could be a more healthy way to manage this disorder. On this graph, you can see the rate of neuropsychiatric disorders before and after lockdown, the blue bar being pre-COVID-19 and the yellow bar being from lockdown. Now, the asterisk above depression means that there is more than a 95% significance for that specific disorder, and it really shows that there is a very high increase in people with depression. Now, why this is, it could be loneliness, it could be other things like lack of basic needs, but nonetheless, there is an increase in neuropsychiatric disorders, especially depression. So finding a medication or finding some type of treatment plan that can help deal with these problems and be not as invasive as current medication would be amazing. Now, let's dive a little bit more into the article that we're going to be discussing today. Previous studies have found that the microbiota of the human gut impacts outcomes of neuropsychiatric disorders in humans. But this study currently wants to determine whether inflammatory Escherichia coli 1 and anti-inflammatory Lactobacillus mucosae NK41 have specific impacts on the neuropsychiatric behaviors of mice when introduced to them. Now, these microorganisms are isolated and then introduced to the mice, and the mice are presented to a battery of tests which show or determine whether these mice are presenting any neuropsychiatric behaviors. Now, what this study specifically wants to determine is whether Lactobacillus mucosae can reduce the altered cognitive function and depression that is caused by Escherichia coli. Escherichia coli has already been shown to cause cognitive decline and depression in mice due to inflammatory cytokines. Now, let's discuss a little bit about the methodology when it came to infecting the mice and then measuring their neurocognitive function after infecting them. Now, K1 and NK41 were isolated from a healthy human microbiome and then cultured in general media. This was so that they could infect the specific pathogen-free male mice with K1 at doses of 1 times 10 to the power of 8 and 1 times 10 to the power of 9 colony-forming units through an oral gavage. And this was done over the course of 5 days. Now, some of the mice that were infected with K1 were then treated with NK41 at doses of 1 times 10 to the 9 colony-forming units. Now, there were two types of tests when it came to testing their cognitive abilities. The first test was based on immobility, the tail suspension task, in where mice were held by their tail and the amount of time measured of them struggling to get out of this hold was measured. An EMP task, which was a maze, and the amount of time it took for the mice to give up and stop moving was measured. And then a forced swimming task meant how long could the mice swim before it gave up and stopped moving. Now, the other type of cognitive test was based on the mice's movement. It measured how long it would take for the mice to get out of a Y maze, or how long it would take for a mice to recognize a novel object, and how long would a mice take to get out of the Barnes maze. When it came to DNA extraction and sequencing, bacterial DNA was extracted from fecal samples, and they chose to sequence this bacterial DNA using Illumina ISEQ 100 sequencing technology. So they chose Illumina because Illumina has short read lengths and a high throughput. They only wanted to sequence the V4 region of the bacterial RS16S rRNA gene, which means that this was a very short sequence. Now, Illumina is not good at reading entire bacterial genomes, but it is very good at giving you a short sequence when you want to find one. And in this case, they just want to see the abundance of a specific bacteria inside of the fecal sample. Now, they were looking for E. coli and L. mucosae, and Illumina gave them all the power they needed to be able to do this sequencing cheaply. So the bioinformatics they used was they sequenced groups into operational taxonic units, and then they analyzed functional genes using PICRUST, or phylogenetic investigation of communities by reconstruction of unobserved states. In layman's terms, this means that they were just looking for what the bacteria inside of a mice's microbiome was trying to do, whether it resulted in a positive effect or a negative effect. This gave them an idea of what their microbiome might look like and what type of microorganisms might be in there. Let's move on from the methodology of the last section and begin talking about the results of the experiment, specifically what behavioral changes researchers saw after the mice were treated with K1 and NK41. Now, mice treated with doses of NK1 had worsening results for the following tests: the Y maze, the elevated plus maze, the forced swimming task, the tail suspension task, and the Barnes maze task. This shows that K1 is a possible factor in worsening neuropsychiatric behaviors, as all of these mice did worse in these tasks. Now, mice treated with doses of NK41 had no effect on the following tests: the Y maze, elevated plus maze, the forced swimming task, and the Baines maze task. This shows that NK41 does not affect neuropsychiatric behaviors in a negative way. Now, let's talk a little bit more about how K1 could have possibly created these neuropsychiatric behaviors. First, the K1 treatment resulted in an infiltration of IBA1 plus cells, NF-kappa B activation, BDNF suppression, and Krebs phosphorylation suppression in the hippocampus, while NK41 had no effect on these factors. Specifically, I want to talk about NF-kappa B activation. This is responsible for activating pro-inflammatory genes. They are transcription factors that activate pro-inflammatory genes and could cause brain inflammation, specifically in the hippocampus, as you can see on the left there. it could be a very real reason as to why there were worsening neuropsychiatric behaviors. Now that we've talked a little bit about inflammation in the hippocampus, let's talk a little bit more about inflammation in the gut. Now, mice treated with K1 were found to have increased tumor necrosis factor, or TNF-alpha expression, and NF-kappa B activation in macrophages. As previously discussed, NF-kappa B is a gene transcription factor that activates pro-inflammatory genes. Tumor necrosis factor is a cytokine that can activate pro-inflammatory responses. This means that when mice were treated with K1, there was a lot of inflammation in their gut, which could cause gut dysbiosis. Now, mice treated with NK41 did not see an increase in either of these factors, but when mice were treated with both K1 and treated with NK41, uh, they found that NK41 potentially suppressed tumor necrosis factor expression and the activation of NF-kappa B in macrophages. Let's talk a little bit more about tumor necrosis factor and the concentration within the gut when comparing it to control groups, K1 treatment, and NK41 treatment. The graph on the left shows the concentration of tumor necrosis factor. The first bar indicates a control group, so neither K1 nor NK1 was treated. Now, we can see right next to that control group is two groups treated with colony forming units of 10 to the power of 3 and 10 to the power of 5 of NK41. We see there is no effects on the amount of tumor necrosis factor expression. But once we begin to treat mice with K1, we can see that treating mice with colony forming units of 10 to the power of 3 causes an increase in tumor necrosis factor when it's K1, and increasing the colony forming units to 10 to the power of 5 also increases the amount of tumor necrosis factor expression. Now, the graph next to it shows the difference between the control group, mice treated with only K1, and then mice treated with both K1 and NK. We can see that there isn't much of a difference between the mice treated with 10 to the power of 3 NK41 and those treated to 10 to the power of 5 colony forming units of NK41. But there is a little bit more variation for the 10 to the power of 5 NK41. The point being here that mice treated with NK41 could saw a reduced expression in tumor necrosis factor even if they were treated with K1 before then. So I previously talked about gut dysbiosis, but let's talk a little bit more about how the microbiome composition shifts once mice are treated with either K1 or NK1. Now, mice treated with K1 showed reduced microbial diversity compared to the control group, but unexpectedly, mice treated with NK41 showed reduced microbial diversity as well. Now, those mice that were treated with K1 showed an increased abundance of protobacteria and enstenobacteria, and a lower abundance of bacteroidetes and vercumicrobia. Now, those treated with NK41 showed a higher abundance of verrucomicrobia, and they showed a lower abundance of protobacteria. Why does the microbiome composition of the mice treated with NK41 and K1 matter so much? Well, we don't want to cause gut dysbiosis, and we can use this graph on the right to kind of show how badly this gut dysbiosis would be caused. First, let's compare the control group to the mice treated with just K1. Here we can see that the phylums, which are stated here on the right, and their colors are here too, we can see that the phylums are significantly different. It seems like in the K1 group, there's significantly less bacteroidetes, which are very important in metabolism and have other functions. Basically, we need those. Now, although I previously mentioned that the microbial diversity in the mice treated with NK41 is decreased. Once we look at this graph, we can kind of tell that it's not too bad. Let's compare blue to blue, red to red. It's about the same here. The biggest difference here is the green. There's a lot more green, meaning there's less protobacteria inside the group of NK41. This is significant enough to mention, but we're not harming the microbiome significantly when taking NK41 treatment. Now, something I failed to mention in the last slide was the abundance of E. coli and L. mucosae in the gut microbiome before, after, and when it's treated together. So first, let's look at E. coli. E. coli has an abundance about there. You can see the graph. Now, when we look over at when it's treated with NK41, we can see that L. mucosae does reduce the amount of E. coli, and K1 significantly increases it, which is expected. But we can see that when K1 is used to treat mice, followed by NK41, we can see that there is a significant dip in the abundance of E. coli, meaning that NK41 is making a difference in restabilizing the gut microbiome. We can see that it kind of brings it back to normal. It's about the same between control group and the group treated with both NK1, NK41, and K1. Now, when we look over at the abundance of L. mucosae, we can see that in the control group, it's about 1, NK41 raises it, those treated with K1 lowers it, and we can see that when it's treated with both, it brings it back to normal. Maybe a little bit higher than the control group, but this is more similar to the control group than anything else. So the use of L. mucosae can disrupt the gut dysbiosis that K1 treatment causes. Now that we've talked about these topics separately, it's time to bring it all together and talk about the correlation between the microbiota and the neuropsychiatric behavior in mice. Now, overall, a higher abundance of K1 was linked to an inflammatory response and worse behavioral outcomes. What does this mean? K1 resulted in inflammation in the hippocampus, inflammation in the guts, and resulted in worse scores for cognitive testing versus the control group mice. Now, NK41 was shown to be correlated with improving behavioral outcomes and inflammatory responses in mice that were already infected with K1. What does this mean? Mice that were only treated with NK41 did not receive any different inflammatory response or any different cognitive test results from the control group. But if a mouse was infected with K1 and then treated with NK41, then there was an improvement in behavior and an improvement in inflammatory responses. In this visualization from the article, we can kind of see what's going on, right? First, let's start from the left, the bottom left, where it says inflammatory K1. We can see that there's gut dysbiosis occurring here, an increase in specific phylums and a decrease in some. And we can see that in the colon, we're experiencing very specific inflammatory cytokines, tumor necrosis factor, and gene transcription factors, NF-kappa B, increasing inflammation in the colon. Now we can see here that anti-inflammatory NK41 is having a couple of different effects, right? It's trying to decrease this NF-kappa B in the increase of gene transcription factor in the brain to stop the hippocampus from having inflammatory response. And it's trying to stop inflammatory responses in the blood as well as the colon. But these arrows showing up and down are showing what the inflammatory K1 is doing, and then when you see these lines, it's basically showing that this NK41 is trying to do the exact opposite, or trying to decrease these up arrows or trying to increase these down arrows. Now, the key findings from this is that E. coli does exacerbate neuropsychiatric symptoms, and that L. mucosae can mitigate these neuropsychiatric symptoms. But there are some limitations in this. Although we know that E. coli exacerbates neuropsychiatric symptoms in humans for sure due to other studies, there is no study that currently says that L. mucosae mitigates neuropsychiatric symptoms in humans. This means that we need to figure out a way that this study can translate to humans. However, that's still an issue because the mechanisms of L. mucosae are still not fully understood, so more research needs to be done on how L. mucosae functions in animals and then in humans before this study can be translated over to a human population. In conclusion, this study provides even more evidence that the microbiome does impact neuropsychiatric symptoms, but more importantly, it shows that L. mucosae NK41 is a promising antibiotic with anti-inflammatory properties that could possibly serve as a more natural and less invasive form for neuropsychiatric symptom intervention. And it could be something that could help the world. We've previously discussed that there's an increase in neuropsychiatric disorders post lockdown, and that's something that people are still feeling in 2025, even though that was almost 5 years ago now. These are 5 questions I put together based on this presentation that might help you understand the connection between the brain and the gut more, and might help you understand how bacteria are so important in our mood, emotions, and how they make neurological compounds and how they can help us deal with our neuropsychiatric disorders. These are the works cited for this presentation. Thank you for listening and have a nice day.